Today we're going to discuss the different biological molecules. Well, what are biological molecules? All living things are made up of four classes of large biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Within cells, small organic molecules are joined together to form larger molecules. Macromolecules are large molecules composed of thousands of covalently connected atoms. Well, what are these things? A polymer is a long molecule consisting of many similar building blocks. These small building block molecules are called monomers. 3 of the 4 classes of life's organic molecules are polymers: carbohydrates, proteins, and nucleic acids. Putting together and taking apart. This is going on constantly inside of our bodies. A condensation reaction, or more specifically a dehydration reaction, occurs when 2 monomers bond together through the loss of a water molecule, hence the name dehydration, loss of a water. Enzymes are macromolecules that speed up the dehydration process. Polymers are disassembled to monomers by hydrolysis, which is a reaction that is essentially the reverse of a dehydration reaction. There are 4 main macromolecules. Each cell has thousands of different kinds of macromolecules. Macromolecules vary among cells of an organism, vary more within a species, and vary even more between species. So we, as humans, do not have the same macromolecules as, say, a lizard, or even more diversity amongst a human and, say, a bacteria. An immense variety of polymers can be built from a small set of monomers. Carbohydrates include sugars and the polymers of sugars. The simplest carbohydrates are monosaccharides, or simple sugars. Carbohydrate macromolecules are polysaccharides, polymers composed of many sugar building blocks. Monosaccharides have molecular formulas that are usually multiples of CH2O. Glucose is C6H12O6, and that's the most common monosaccharide. Monosaccharides serve as a major fuel for cells and as raw material for building molecules. A disaccharide is formed when a dehydration reaction joins 2 monosaccharides. So as you can see, 2 beta molecules, a galactose and a glucose, come together, a dehydration reaction occurs, and you get lactose, which is milk sugar, the sugar that is in milk and ice cream. Polysaccharides. These are really important in biology, and we're going to learn why. Starch is a storage polysaccharide of plants, and it consists entirely of glucose monomers. So potatoes contain a lot of starch, and they hold a lot of energy for our cells. When you eat the potato, your body is able to break it down and deliver the energy in the form of those glucose monomers. Plants store surplus starch as granules within their chloroplasts and other plastids. So inside the plant, they actually store this extra polysaccharide material. Glycogen is a storage polysaccharide in animals. Humans and other vertebrates store glycogen mainly in our liver and muscle cells. And the polysaccharide cellulose is a major component in the tough wall of plant cells, which we'll learn about later this year. So as you can see, a leaf, up close and personal, and then up close even more personal with those cellulose microfibrils in a plant cell wall. And if we took one of those microfibrils apart, you'd break it down to all those little tiny cellulose molecules and eventually down to a glucose monomer. Here's some neat random facts about polysaccharides. Cellulose in human food passes through the digestive tract as insoluble fiber. So if it weren't for the bacteria in our digestive tracts being able to break down that insoluble fiber, we wouldn't be able to digest any fruits or vegetables. Some microbes use enzymes to digest the cellulose. And many herbivores, from cows to termites, have symbiotic relationships with these microbes. Did you know that if it weren't for the microbes and the bacteria inside the termites' digestive tract, termites would not be able to digest the wood? Pretty neat. Chitin is another structural polysaccharide, and it's found in the exoskeleton of arthropods. Chitin also provides structural support for the cell walls of many fungi. So as you can see, there's the structure of chitin, the monomer, and it's showing that chitin forms the exoskeleton of arthropods, and it's used to make strong, flexible surgical thread. Pretty neat. Lipids are the one class of large biological molecules that do not form polymers. The unifying feature of lipids is having little or no affinity for water. They're said to be hydrophobic because they consist of mostly hydrocarbons, which form nonpolar covalent bonds. And the most biologically important of these lipids are fats, phospholipids, and steroids. Well, take a look at the lipid structure. Fats are constructed from two types of smaller molecules, glycerol and fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl, that's the OH group, attached to each carbon. See that molecule on the left? That's glycerol. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. This is why oil and water don't mix. The fats separate from water because water molecules form hydrogen bonds with each other and exclude the fats. In a fat, 3 fatty acids are joined to a glycerol by what's called an ester linkage, creating a triacylglycerol or triglyceride. You may have heard that triglycerides are not good for your heart. Well, there's different types of fats, and we'll get into those as well. The fatty acids vary in length, mainly by the number of their carbons, and in the number and locations of the double bonds. Saturated fatty acids have the maximum number of hydrogen atoms possible and no double bonds, whereas unsaturated fatty acids have 1 or more double bonds. So as you can see, the saturated versus the unsaturated. Notice the double bond? Unsaturated fatty acids act as anticoagulants in the bloodstream, unlike saturated fatty acids, which tend to make your blood sticky for hours after a fatty meal. So think of this next time before you head to your favorite fast food joint. A diet rich in saturated fats may contribute to cardiovascular disease through plaque deposits. We just said how it makes your blood thicker for hours after a fatty meal. Hydrogenation is the process of converting unsaturated fats to saturated fats by adding hydrogen. So if you look on the back of your country crock butter spread or whatever you use as a buttery spread, sometimes you'll see where it says partially hydrogenated vegetable oil. That's what they're talking about. Hydrogenating vegetable oils also creates unsaturated fats with trans double bonds. These trans fats may contribute more than saturated fats to cardiovascular disease. So notice a lot of times now, especially even at McDonald's, you'll see they advertise no trans fat in our oil that we use to fry all of our French fries. Well, fats actually do have a good function, and they are a main source of energy storage. Humans and other mammals store their fat in adipose cells, which is the scientific way of saying fat cells. Adipose tissue also cushions vital organs and insulates the body. Fats made from saturated fatty acids are called saturated fats, and they're solid at room temperature. Most animal fats are saturated. So there's your wonderful dish of butter, and that is from saturated fat because they are solid at room temperature. Fats made from unsaturated fatty acids are called unsaturated fats or oils, and they're liquid at room temperature. Plant fats and fish fats are generally unsaturated. So we're talking about your peanut oil, corn oil, vegetable oil, olive oil. Those are all unsaturated fats. In a phospholipid, 2 fatty acids and a phosphate group are attached to a glycerol. These 2 fatty acid tails are hydrophobic, which means they do not like water, but the phosphate group and its attachments form a hydrophilic head. This becomes really important when we start talking about the structures of our cell walls, and we'll get into the cell membrane structure later on when we talk about cells. When the phospholipids are added to water, they self-assemble really neatly into this little double layer called a bilayer, with the hydrophobic tails pointing toward the interior. The structure of phospholipids results in a bilayer arrangement found in cell membranes. and phospholipids are the major component of all cell membranes. Now let's discuss steroids and cholesterol. Steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. Cholesterol is an important steroid, and it's a component in animal cell membranes. Although cholesterol is essential in animals, high levels of cholesterol in the blood can contribute to cardiovascular disease. Let's discuss proteins now, because proteins are really important and they account for more than 50% of the dry mass of most cells. Proteins' function include structural support, storage, transport, cellular communications, movement, and defense against foreign substances, such as antibodies. If you've ever heard of them with your immune system, antibodies are actually a protein. We're also going to discuss enzymes, and they're an important biological molecule because they're a type of protein that acts as something called a catalyst to speed up chemical reactions and make them go faster. Enzymes can perform their functions repeatedly, functioning as workhorses that carry out the processes of life. Polypeptides. Poly, of course, means many, and peptides, we're talking about proteins. Well, polypeptides are polymers built from the same set of 20 amino acids. Remember, amino acids come together to build proteins. A protein consists of one or more polypeptides. We just talked about amino acids. They're the organic molecules with carboxyl and amino groups. So notice that carboxyl, the C-double-O bond and the OH, that's a carboxyl, and then the amino group on the left side is the N with the 2 H's. The amino acids differ in their properties due to differing side chains. So notice that R group attached to the carbon, that is the differing side chain that characterizes each amino acid. So amino acids are linked by peptide bonds. A polypeptide is a polymer of amino acids. Polypeptides range in length from a few to more than 1,000 monomers. Each polypeptide has a unique linear sequence of amino acids. The sequence of amino acids determines the protein's three-dimensional structure, and a protein structure then determines its function. So check this out. The abdominal glands of the spider secrete silk fibers made from a structural protein containing pleated sheets. The radiating strands made of dry silk fibers maintain the shape of the web, and the spiral strands, those are the capturing strands, those are elastic and they stretch in response to wind, rain, and the touch of insects. Pretty neat. So what happens if protein structure changes? Well, a slight change in primary structure can affect a protein's structure and ability to function, such as sickle cell disease, which is an inherited blood disorder. It results from a single amino acid substitution in the protein hemoglobin. So on the left, you'll see a normal red blood cell, which is full of individual hemoglobin molecules, each carrying oxygen. On the right, you'll see that sickle cell shaped with the red blood cell, where the fibers of abnormal hemoglobin deform the red blood cell into a sickle shape. This can cause major health problems for the person affected by sickle cell anemia. Finally, we have to talk about nucleic acids, because without them, we would not be here. The amino acid sequence of a polypeptide is programmed by the unit of inheritance called a gene. We all have genes, from us as humans all the way down to bacteria, the simplest forms of life. Genes are made of DNA, which is a nucleic acid. There are 2 types of nucleic acid. One I'm sure you've heard of before, deoxyribonucleic acid, or DNA. The other we may not have heard of. It's called ribonucleic acid. You may not have heard of it, but it's actually just as important as DNA. They work together. DNA provides the direction for its own replication, and DNA directs the synthesis of something called RNA, the messenger RNA, and through messenger RNA controls protein synthesis. Well, that's all, folks. Just a quick review of all of the 4 biological molecules, which are very important in the study of biology.